Thank you, and thank you everyone for being here today. So my talk today is about various neuroimaging software that helps to unite structure and function in human MRI research. You might notice that the word tutorial is in my talk, and this is a 25-minute talk. Sorry, is in my title, and this is a 25-minute talk. So I'd like to encourage you to think of this a little bit more like a software overview. I will include some demonstrations, but if you are interested in tutorials, I have gathered a bunch of tutorials for the software I'm going to talk about today in a GitHub page here on the screen. And in fact, I'm going to keep that GitHub page in the lower left corner of my slides for this entire talk, so you can find it easily. I highly encourage you to go to this GitHub page if you're interested in any of the software I talk about. The tools in this talk are of interest to neuroimaging research generally, but I study visual neuroscience as a domain, so I'm going to start by giving a very brief introduction to human vision. So shown here are the two inflated cortices of a human brain, but to make visualization a little easier, I'm going to just ignore the left hemisphere, and we'll focus on the right hemisphere. Now the gray trapezoid shown on the left represents a stimulus screen during a functional MRI experiment, and the white dot indicates the position at which the subject is pointing their eyes during the experiment. Here I've put a little bit of stimulus at the center of the screen near the subject's center of gaze. And I've colored the cortical surface by its bold response to the stimulus. Because we're looking only at the right hemisphere, and because the right hemisphere receives visual input only from the left side of the visual field, I've limited the stimulus to just the left side of the screen. Now, with the stimulus in the center of the screen, the cortical activation mostly occurs in the posterior cortex. However, if we sweep the stimulus out to the subject's periphery and back, the region of cortical activation will sweep towards the brain's anterior and back in a very clear pattern. If we were to take this pattern and visualize it as a single image, it would look something like this. Here, the stimulus is colored according to the distance from the subject's center of gaze, and the cortex is colored according to the part of the visual field where a stimulus produces a maximal response. So, for example, I don't seem to have a pointer, but for example, the posterior blue part of the cortex responds when the stimulus is in the blue region of the screen. This overall property is called the visual eccentricity. Additionally, we can measure the pattern of response when the stimulus sweeps in the orthogonal direction, that is, around the visual field. This stimulus induces a number of stripes of cortical activation that sweep simultaneously across cortex. Each independent stripe in this case represents a different visual area, and if we visualize these as a single property like the eccentricity, it looks like this. Here, cyan color corresponds to the upper visual field, and the reddish colors correspond to the lower visual field. This property is called the polar angle. An important feature of the organization of the visual cortex that we can see from these maps is something called the cortical magnification, which is actually a direct measurement of the relationship between the structure and the function of the brain. To explain this, let's look back at the eccentricity map briefly. Notice that the blue region on the cortex covers almost half of the visually active cortical surface, yet the blue region in the stimulus covers only a small fraction of the visual field. Inversely, the orange color that represents the periphery takes up a large amount of the visual field, but a relatively small amount of cortex. This is because the center of gaze is magnified relative to the periphery on the cortical surface. Calculating the cortical magnification requires a pretty good model of how the visual field is laid out on the complicated 3D cortical surface. That is, a model of the relationship between structure and function itself. And we will work up to that model in these slides. Before we get there, however, I will start out by pointing out that this measurement, cortical magnification, correlates well with visual acuity in individual subjects. And in fact, if you can calculate it very carefully, it correlates in individual points in the visual field with subjects' acuity at those individual points. Now, individual differences in the relationship between structure and function, such as those described by the cortical magnification, are one of the many reasons I find human vision so interesting. What you're seeing here is an animation of 3D brains that are morphing iteratively from one subject into the next and into the next, pausing briefly on each individual subject, for all the subjects in a dataset called the New York University or NYU Retinotopy dataset. The brains are colored according to each subject's polar angle response to functional MRI experiments. Simultaneously, the plot of the visual field at the bottom of the slide shows each subject's cortical magnification, where dark colors represent a low magnification and lighter colors represent a higher magnification. At first, this may just seem like kind of a fun just kind of a fun animation, but I also intend for it to demonstrate some important things about the relationship between structure and function in the visual cortex. First of all, it should be pretty clear that there is an enormous amount of variability in the shape and size of the visual cortex. In fact, if you look at the Cochrane sulcus, which usually I would point to at this point, but it's basically the great big groove around which all of the color that we're looking at is. If you look at the Cochrane sulcus, it's basically where the first visual area, V1, lives, and that visual area can vary by a factor of over 3.5 between subjects. Simultaneously, the entire cortical surface can only vary by a factor of about 1.5 between subjects. Despite this enormous structural variability, however, the general organization of the visual cortex is highly conserved, with the upper bank of this large sulcus in the back of the brain always representing the lower visual field, and the lower bank always representing the upper visual field. Unsurprisingly, these variations in structure lead to large variations in the cortical magnification as well. Making this animation that I just showed you requires a lot of software tools. For starters, someone had to run the actual MRI experiments and collect the raw data. And while this step is incredibly important for any neuroscience research, this talk is a software talk, so I will be focusing on the steps that follow the data collection. Once the data were collected, they had to be pre-processed, which in this case was performed using the tool fMRIprep. And fMRIprep itself calls a pre-processing tool called FreeSurfer, which extracts and calculates structural information about each subject. Once the data have been pre-processed, it was saved to a common file format called BIDS, which stands for the Brain Imaging Data Structure, then uploaded to a neuroscience dataset repository called OpenNeuro. Finally, I had to actually download the data, model it in order to calculate the cortical magnification, and render it as a bunch of movie frames. And for these steps, I used the repository access tool DataLad and my own Python library, neuropythy, for modeling and rendering the frames. These steps will basically form the outline for the remainder of my talk, during which I will step through these tools and provide an overview of each. And as a reminder, if you're interested in any of these tools, there are more resources and tools available at the GitHub page that I have at the lower left corner of these slides. We'll start by discussing some of the data processing tools used for this dataset. The NYU Neuroanatomy dataset from which the animation we just saw was generated was pre-processed using the tool fMRIprep, which I highly recommend due to its ability to standardize functional MRI pre-processing steps. This tool is one part of the neuroimaging preps community, or NEPREPs, and in fact fMRIprep is one of the main entry points for its software tools. fMRIprep automates most of the structural and functional pre-processing of raw MR images. It handles details such as slice time correction, structure function alignment, motion correction, resampling of functional data into structural spaces, and additionally it outputs the data in BIDS format, which we're going to talk about in just a second. fMRIprep calls many other tools, including FreeSurfer and MRIQC, which is a quality control tool for MR images. And these tools generate a number of reports in web page format so that researchers can easily see the quality of their data and pre-processing steps. The images shown here are examples of these quality control reports that are related to structural processing. Structural preprocessing itself is usually performed by FreeSurfer, or at least I usually perform it by FreeSurfer. FreeSurfer is a very mature tool that was developed back in the 90s and has gradually assumed a critical role in contemporary neuroimaging work. FreeSurfer performs a great number of usually tedious tasks in the structural preprocessing pipeline, such as brain alignment, removal of the skull, gray-white matter segmentation, surface tessellation, cortical inflation, and anatomical atlas alignment. The first many steps in the FreeSurfer pipeline involve identifying and describing the cortical surfaces. We don't have time in this talk to go into these steps in detail, but the basic idea is that FreeSurfer converts a 3D voxel-based image of the brain, such as the one you're seeing on the left, into 3D models of the brain's cortical surface, such as those shown on the right, using a series of finely tuned filters, heuristics, and algorithms. Another important utility performed by FreeSurfer is anatomical alignment. Now, in the animation I showed earlier, one of the key things that we would need to know in order to make that animation is how to morph one subject's brain into that of another. Doing this requires an understanding of how any given point on the first subject's cortical surface needs to move in order to land at the correct point on the second subject's cortical surface. In other words, we need the cortices to be aligned according not just to rotation and translation, but in terms of the folds at the cortex itself. The idea behind FreeSurfer's anatomical alignment is to normalize away the many differences in anatomy between subjects. For this process, we start by collecting structural MR scans, such as those shown here, from many subjects, these days typically more than 1,000 subjects. We then average their MR images together, voxel-wise, producing this image of a very smooth cortex. Now notice that the individual brains have a lot more folds and wrinkles in them, while the average brain captures roughly how an individual brain might be shaped. We can then extract a 3D model of the brain from this representation. And as an aside, we can actually make these 3D models from any structural MRI scan. And they are basically represented as triangles and vertices, just like modern 3D computer graphics. Next, we take this 3D model of an average hemisphere, which we sometimes call an atlas hemisphere, and is shown on the left, along with the model of an actual person's brain, shown on the right, and we can computationally inflate them. Inflation basically makes these hemispheres easier to work with because it turns them into spheres, and spheres are geometrically simpler. Once inflated, we might be able to see that there's some correspondence between the pattern of curvature indicated by the color map here, but the correspondence is not exact. So, for example, if we highlight the calcarine sulcus in the atlas hemisphere, that's the highlighted sulcus here, and we transfer that region over to the identical position on the individual hemisphere, we can see that it doesn't line up very well with the curvature of the individual. FreeSurfer corrects for this by treating the individual subject's cortical surface like a big rubber sheet that can be stretched and compressed but not torn or folded. By stretching and compressing the individual hemisphere's surface, FreeSurfer finds the optimal correspondence of curvature between the two hemispheres. This process looks a little like this. So as we can see, after this registration, the calcarine sulcus of the individual subject is well aligned to that of the atlas, and this is true for other sulci injuri as well. Overall, this creates a correspondence between the points on the subject's cortical surface and the surface of the atlas hemisphere. So if we then get another subject and align their hemisphere to the same atlas, we will have a correspondence of points between those two subjects, which allows us to morph one subject's brain into that of the other. This is also the method by which standard atlas models, such as the Wong 2015 probabilistic atlas of brain areas, are able to transfer an atlas over to an individual subject via this alignment. Once the data from visual experiments has been pre-processed, it would typically undergo a set of analyses in which we would solve what are called population receptive field models, or PRF models for each voxel. These PRF models result in the polar angle and eccentricity maps that I showed you earlier. However, this is all outside the scope of this talk, so we will instead assume that these PRF models have already been solved by vision experts, and we'll move along to data sharing. One of the most significant advancements, I think, in the sharing of neuroimaging data in recent years is the development of the Brain Imaging Data Structure, or BIDS. BIDS is a system for organizing both the raw data and the derived data from neuroimaging experiments such that they can easily be understood and interpreted by others. BIDS is an open, self-governed standard that handles not just MRI data, but physiological, genetic, and behavioral data, and it works for many modalities, not just MRI, but also MEG, EEG, electrocorticography, PET, and microscopy. The image on the right here shows how a typical directory of machine-named DICOM files would be transformed into a set of files with standard formats for BIDS that includes metadata about the experiments. Such an organization is valuable in part because it enables both humans and computers to easily read and understand the data, thus enabling computational analyses to operate across many data sets. These features make BIDS critical to the sharing of data, and in fact, the website OpenNeuro, which hosts the NYU Retinotopy dataset, that was used to make the animation earlier, is specifically designed for researchers to deposit and share BIDS datasets. OpenNeuro is free to use as a repository of BIDS data, and datasets deposited there are freely available to others. The website gives each dataset a unique identifier that can be used to look up its data and metadata. The screenshot on the right of this slide actually shows the information page for the NYU Retinotopy dataset, which has the identifier DS003787 at OpenNeuro. The dataset can be downloaded directly from this website. You can also get it using Amazon's S3 service or using tools like DataLad. This brings us to the final set of topics in my talk today, tools for accessing and manipulating the dataset. So we'll start with the DataLad tool. DataLad is a simple command line tool that's very similar to Get, but instead of interacting with code repositories, it interacts with data repositories. DataLad can be easily installed on any operating system by following instructions on its website, datalad.org. I'm now going to attempt a demo here using DataLad to download the NYU Retinotopy dataset. So hopefully here you're seeing a terminal window for my laptop. To start with, I'm going to go ahead and switch directories to a location on my laptop where I can store OpenNeuro datasets, which for me is just /data/openneuro.org. I will then run the command datalad clone, which is similar to the git command for cloning repositories from GitHub, and I will provide it with a GitHub repository which contains the metadata for the NYU dataset. You can see it ends with DS003787, which is the identifier. All Open Neuroset datasets can actually be downloaded in this basic way, and you'll notice that the download is rather fast. In fact, they're just finished. This is because only the BID's metadata and directories have been downloaded while the data files themselves have not. We can now see that there is a directory for this dataset here, and if we traverse into it, we'll be able to see that it contains a directory for each subject along with standard BID's metadata. For example, the participants.tsv file, which you should be able to see there, is a tab-separated value file that contains a list of basic information about participants, so we'll go ahead and just take a look at that. There you can see all the various participants, their ages, gender, etc. To make the animation we saw earlier, we would need to at least get data from the derivatives directory, which stores processed as opposed to raw data. So for example, we can see in the derivatives directory here that there is a directory for FreeSurfer as well as directories for example, ROIs that are hand-drawn, or there's a directory here called PRF analyze-vista, which actually contains the visual data we saw earlier. We'll go ahead and take a look at the FreeSurfer directory. And as you can see, there's this directory contains a directory for each subject, and each subject's FreeSurfer data is contained inside those directories. That said, the data files themselves haven't actually been downloaded yet because all we ran was datalad clone, so we need to go ahead and get these data. To do that, we issue a command datalad get, and we give it a list of directories or files that we want it to actually download. So here we'll go ahead and tell it to download the FreeSurfer directory, the ROIs directory, and the PRF analyze-vista directories, all of which are in the derivatives directory. And datalad will go ahead and begin downloading those diligently. And now you may notice that there's about 20 gigabytes to download, so this is going to take a while. We're not going to wait for it. You're just going to have to trust me that it'll finish. Once we've successfully downloaded the data in BID's format, we just need a tool that can load, model, and render it. For this, we will look at my own library, neuropythy, which is a library of utilities for cortical surface analysis that is intended to serve as a sort of glue between structural utilities like FreeSurfer and more complex or nuanced analyses of cortical data. What you should be seeing here is a Jupyter notebook window, which I will use to demonstrate some of what neuropythy is capable of. For starters, I'm going to go ahead and select that input cell, and I'm going to go ahead and import neuropythy with the alias ny. All right, so now I'm going to go ahead and define a few directories. So we'll start with the path for the data. So as a reminder, that was /data/openneuro.org/ds003787 for that data set. I'll then define the derivatives path, since we'll be loading data from the derivatives path, and that's basically in the data path /derivatives. I'll then define the FreeSurfer path, which is again in the derivatives path, so we'll put it in derivatives path /FreeSurfer. And then I'm going to define one more path, which is the PRF path. This is the path that contains the actual visual data. So this is in the derivatives directory /PRF analyze-vista. We saw that directory earlier as well. All right, now I'm just going to define a subject, and we'll just use subject 1, I think, for this example. And then we'll go ahead and define a session, and this just refers to the name of the MR session that was used in this data set to collect the visual data. Okay, so at this point, let's go ahead and ask FreeSurfer to load us. Actually, let's start by looking. Let's look at these paths just to make sure we got them right. So here we can see. Okay, so we can see all the subject directories here, including the one we're going to look at, the derivatives directory. Let's go ahead and look at the derivatives directory as well. Actually, the FreeSurfer directory. So we can see that we've got our FreeSurfer directory here as well. So these paths all look okay. So now at this point, let's go ahead and have neuropythy load a FreeSurfer subject. So for this, we just call the FreeSurfer subject function, and we just pass it the path of the FreeSurfer subjects directory. So for us, that's forgot my bracer there. So that's just the FreeSurfer path followed by the subject's name. And this will go ahead and give us back an object. Let's take a look at it. This is an object called a subject object that knows where this subject is stored on disk. So at this point, let's go ahead and do a visualization. So we're going to use the function neuropythy.cortexplot, which will plot the 3D surface, and we're going to pass it the subject's left hemisphere here. And we're going to ask it to plot the inflated cortical surface to start with. So when we run this, we'll get a 3D model here in the browser that you can actually interact with. This is the inflated surface for this subject, for the left hemisphere. Let's take a look at the white surface as well. So we'll just ask it to plot the white surface here. And that's a much more normal looking surface to me anyway. So here we have the white surface, so that seems to work fine. So at this point, let's go ahead and load some of the visual data. So we'll start by loading in the polar angle data. And for this, we're going to use the neuropythy.load function, which knows how to load in most neuroimaging formats. We're going to pass it the PRF path we defined earlier, followed by the subject's directory, followed by a directory for the session. And then we're going to just give it the name of the polar angle file. NeuroPythy knows how to read most of these files. Let's load the eccentricity as well. That's going to be pretty much the exact same line. We're going to load it from the PRF path /subject/session, and then it's just a different file name. And in addition, we'll load one more file, the coefficient of determination. And this basically is a file that tells us the confidence at any point on the cortex that we have that it's visually active. So these values vary from 0 to 1, with 0 indicating that that part of cortex is not very active and 1 indicating it is. All right, so let's look at the polar angle here. And what you can see is we've just loaded in a vector of numbers in a NumPy array. And in fact, if we look at the shape of it, there's about 200,000 vertices, and that matches the number of vertices in the subject's left hemisphere, which we can see here. Now, if you're confused as to why these match, keep in mind, like I said earlier, these cortices are actually just like modern 3D graphics, they're just vertices and triangles. So if we zoom way in, you can see that this is actually composed of individual triangles and vertices here. There's a triangle, there's a triangle, there's a triangle. So this hemisphere actually has about 200,000 individual vertices, and we've just loaded in a polar angle value for each individual vertex. That's why they match. All right, so let's go ahead and plot some of these data. So once again, we're going to use the cortex plot function. Once again, we'll pass in the subject's left hemisphere. We're going to ask it to plot the white matter surface this time. And we're going to ask it to color it according to the polar angle we just read in. And just like with Matplotlib, we need to give it a color map. There's a built-in polar angle color map. And we need to tell it the minimum and maximum value. So negative 180 to 180 degrees is the minimum and maximum for polar angle. And when we plot this, we should get a plot of the polar angle across cortex. Now, this doesn't look much like the animation we saw earlier because there's a whole bunch of noise over here. So I'm going to go ahead and add a mask. And for that, we're going to use the coefficient of determination that we loaded in. So we'll just put a mask here that says we only want to plot the color where the coefficient of determination is between 0.1 and 1, so where we have pretty reasonable confidence that the cortex is visually active. And that gives us a plot of the polar angle, much like we saw in the animation. Okay. So at this point in the talk, I've gone through just about everything in this animation earlier, or at least I haven't really showed you everything about it, but I've showed you a little about everything in the animation, with the exception of this cortical magnification plot at the bottom. And I mentioned that in order to make this cortical magnification plot, we need a very good model of how the visual field maps onto the cortical surface. So this is also performed by NeuroPythy. Basically, we are going to use the exact same kind of method that we did with FreeSurfer, where FreeSurfer aligned two cortices based on curvature. But in this case, we're interested in a model of the visual field. So we can take a model of the visual field that's already been developed. Here I've just drawn it as a schematic. And we can basically just plop this down on this orthographic projection of the 3D cortical spheres. So that looks a little like this. I've drawn the model now using just lines so that you can see the data underneath it. What we're going to do is we're going to treat the cortical surface once again like a big rubber sheet, and we're going to stretch and compress the cortex. But this time, instead of aligning the curvature, we're going to align the underlying polar angle and eccentricity data to the model. Now, I'm only showing you the I'm only showing you the eccentricity because I can only show you one at a time, but this actually works for both simultaneously. And that alignment looks a little bit like this. So that brings the underlying cortical surface into alignment with the model. And at that point, we can actually just ignore the data underneath and put a prediction based on the model. And we can relax the cortical surface. See if we can get that animation to work. There we go. We can relax the cortical surface back to its original shape, and we'll have a very nice prediction of how the visual field is laid out on the complicated 3D surface of the brain. And now if we want to calculate the cortical magnification, all we need to do is zoom in on a particular subject's cortex. We have a model of how the visual field is mapped onto the cortex. We can draw a little disc on the cortical surface. Here's one that's about 5 square millimeters in size. We can map that disc right into the visual field using our model, and it'll have a size there. Here this one is about 2 square degrees. And then the cortical magnification can be calculated just by doing some division. And we can do that across the whole visual field or across the whole cortical surface. All right, so I've gone over quite a bit today in this talk. If there was one thing I wanted you to remember from it, it's that the combination of structure and function in neuroimaging research leads to a lot of novel insights and measurements of the human brain. Additionally, we talked about structure and functional data processing tools such as fMRIprep and FreeSurfer, which have the power both to automate and to standardize a lot of the processing needed for these kinds of analyses. We talked about BIDS, which is a really critical standard for contemporary distribution of neuroimaging data. We talked about OpenNeuro, which is a free and open access data repository for neuroscience data. We talked about DataLab, which is a powerful tool for dataset management. And finally, we talked about NeuroPythy, which is a useful open source Python library for manipulating and analyzing visual neuroimaging data. That concludes my talk. Thank you for your attention. I'd be happy to take any questions.
Hello, hi. Thanks for the very nice presentation. I have a couple of questions. I'm interested to know, first, I think the pipeline that you showed is like super cool, super simple. Very impressed with DataLab also. You focused on retinotomy. I'm interested to know if this pipeline in the experience that you have was as simple to implement for maybe task-based fMRI data. Usually the data that you download, does it come with already projected contrast on the surface, etc.? And if not, that would be a second question. Does the library that you showed in the end, your library, handle volumic data, or like, how do you do the end? Thanks.
Yeah, and thank you. Yeah, so let's see. First of all, the particular data set I analyzed already had the data projected to the surface, and however, that said, NeuroPythy does handle volumetric data. It is really designed for surface-based data, but it does handle, in particular, interpolation from the cortical surface and back. So if you had a data set that was volumetric, so for example, even if you had a data set that was volumetric that had not been processed with FreeSurfer and you didn't even have cortical surface structures yet, you could just download the data set, run FreeSurfer on it, and then point NeuroPythy at the FreeSurfer and the volumetric files and have it do the interpolation onto the cortical surface. That's all built into the data set, yeah. I think that I might have missed a piece of your question, but if so, just ask it again.
Any other questions on the floor? Yes. much for your talk. I was just wondering, what's the advantage of using bits? Like, does it anonymize the data better?
Yeah, so BIDS, it doesn't actually do anything. It's just a format standard. So the main value, I think, of BIDS, if nothing else, is that it allows you to put your data on openneuro.org for free. If your data isn't in BIDS format, you can't put it up there. And if you have a large data set, it can be really hard to find anywhere you can put it that is open access. Additionally, if you're ever going to send data to someone else, it's just, I think, polite to send it in BIDS, because if you don't, every lab has a cottage industry. Every lab has its own way of formatting its data and naming its data, and nobody ever likes to try to figure out some other lab's way of dealing with data, and BIDS solves all that problem. So I think that's the main advantage of it, really.
I see. And is there any storage capacity for that?
If OpenNeuro has a storage limit, I'm not aware of it. I think you just have to have it in BIDS format, and I think you're supposed to eventually publish a paper related to it, but I think those are the only requirements.
Okay, thank you.
Mm-hmm.
We still have time for questions.
Thank you for the talk. It was just a question about someone who does volumetric data analysis usually, and I wanted to know how easy it is to manipulate this kind of data on Python, because I'm used to just manipulate NumPy arrays, and I see vertices and things like that. I'm like, is it hard? Is it easy to get into, or?
Oh man, that's a hard question, because it's been a long time since I've gotten into it. I've been into it for a long time, I should say. In my experience, there are some big advantages in how you think about the data that come from having it volumetrically organized, in that it's easy to think about slices and space in terms of voxels. It's a lot harder to think about space in terms of vertices, so I think that's the main challenge. In fact, if we were to look at that array of polar angle values that we read in, and we were to plot the individual vertices one at a time, you would see very quickly that there's no particular way that the vertices are organized on the surface. Vertex 1 and vertex 2 may be next to each other in the array of vertices, but they're often on opposite sides of the cortical surface. That's just kind of a reality of how cortical surface data works. But that said, that ends up being a relatively small problem in the long term. And libraries like NeuroPythy include a lot of tools that make a lot of that much easier and much less problematic to deal with. On the flip side, I think that the advantages of the surface are very high, especially if you're interested in anything related to something like distance. Distances in the volume are very misleading, and distances on the cortical surface are much more meaningful, especially for gray matter.
Sorry, it's me again.
No problem.
I'm not sure it's a question, it's much more of a fantasy, but since we have so much open source data at the moment, and a great deal of it is formatted in bits, sometimes I fantasize that we have this web-based viewer where we can look at the surface data that's been projected on the surface, but directly in the browser. Do you think, how far do you think we are from having this?
Yeah, all the technology for that is already available. It's just that no one's written it yet, as far as I know. So I think not very far. I think we're just kind of waiting for an enterprising lab or postdoc or grad student to write that software. And if one of you is interested in doing that in the audience, send me an email and I'd be happy to talk to you about it. But right now this kind of software is mostly in Python and a few other languages.
it can be very drastic because even for we become adult, we can still run juggling, we can just run new skilled movement, but maybe it's not really generalizable to many other systems because other systems can be drastic. So do you have any kind of idea about how much degree of white matter plasticity is similar across different parts of the brain, or maybe motor is a particular place where you can just find plasticity easily?
So studies so far have looked at different systems. So the first system that people actually looked at was hippocampus, so fimbria and fornix areas. As you would expect, hippocampus is a very plastic structure, so people started looking there and they found evidence of that. Sensory areas, so in this case, because it's rodents, when I say sensory areas, I'm talking most specifically about the barrel cortex, which is the system that that senses the whiskers. So it's kind of the equivalent to the human visual system in rodents. I mean, maybe that's a stretch, but yeah, so rodents, you will use their whiskers to move around and to identify their environment, and you will use much less visual cues. I haven't seen, so yeah, of course, the visual system has also been used more recently. I've seen a couple of studies using particularly deprivation models. And auditory models as well. So it seems like, I don't know if the degree of plasticity is different because that's hard to compare, but it seems like there is myelin or white matter plasticity happening at multi-systems level.
Yeah, thank you very much. Yes, please.
Hi, my question is also for Cassandra in terms of the kind of language that we use to describe differences in white matter in the brain. So kind of often when you kind of read about, I don't know, differences people might see in white matter microstructure, people talk about reductions in integrity. So for example, a reduction in FA, that might represent kind of reduced integrity in white matter microstructure. But, I think I've been reading recently about actually maybe that's not an accurate representation of what's going on kind of at a physiological level, especially in terms of crossing fibers, but now also in terms of, the differences you see in, the kind of diameter in axons and other kind of cellular functions. So I'm just wondering, do you guys have any thoughts on the language that we use to describe these differences in white matter when we're reporting them in our research?
Yeah, so people tend to use the word microstructure when they're talking about healthy conditions, and then they also use integrity for maybe more, non-healthy conditions. Both are umbrella terms, so people use them interchangeably. So I mean, language is really important, but in this case, to be honest, I mean, because we don't know what we are measuring, I would say I'd like to use microstructure, so alterations in microstructure in white matter. That won't tell you anything much because, again, we don't know what is changing exactly, right? Integrity, I've seen it more when people are referring to pathological conditions like MS or stroke, where integrity is affected. If people might associate integrity maybe with axonal damage, but again, we don't know what's measuring, so it can be anything. We could be more precise, but because the technique itself is not really precise, I'm not sure which umbrella term is best, to be honest. But yeah, I think you can always explain the caveat in your research paper that this is an unspecific measure. And so it can be modulated by all these different things, and give a couple of examples just to make sure that people understand you're not saying this is myelin or these are axons.
Great, thank you.
Any other questions from the floor to any of the speakers?
Yeah, if you go up to the microphone.
Hi there. So this is just a general diffusion weighted MRI technique. So usually in diseases, we see the FA increases and, I think the FA decreases and the MD increases in diseases. So what if we see the opposite in a disease? So why do you think we might see the opposite, like a decrease in MD and increase in FA?
Okay, so specifically MD. MD is just a measure of how restricted the water is regardless of direction, right? So for instance, in any tissue really, if you have more water or if you have tissue damage, or if basically you have no restriction due to changes in for instance, fibers or the density of fibers, and I'm talking even the cortex because you can use that MD in the cortex, then you will have increase in MD, right? So thinking of pathological conditions where you can have the opposite, potentially in areas of crossing fibers where you have maybe one of the fibers being degenerated, then you can have that effect. And then an FA would have the opposite effect, obviously. Does that answer your question?
Yeah, sure. Thank you.
Could you come closer to the microphone, or maybe it's not switched on?
Can you, oh, yeah, there we go. So I guess this is a little bit related to the last question. Have any of you, maybe those more familiar with white matter, heard of the eye condition of, it's actually called myelinated nerve fiber, where there's too much myelin on the nerve fibers in the eye, yeah? I'm just curious, if you have any ideas of why too much myelin would cause visual problems. Generally, I mean, I don't work at this level, so I'm not as familiar with the research, but generally I thought more myelin would help the signal. I'm not sure whether it physically blocks the light going into the retina or if it somehow disrupts the signal along its path.
No, I don't know that condition specifically, so I don't know if it is blocking the light coming through the retina, but myelin in general, it will speed up transmission, right? So if you have too much myelin or too little myelin or an alteration in myelin, you're changing the circuit, right? So you might basically cause desynchronization of the signal instead of synchronization of the signal. So if two signals have to arrive to a particular brain area at the same time, and now you're altering myelination on one side and not the other, then they're not arriving at the same time anymore. So these things have to be fine-tuned. How the brain does this specifically when it comes to myelin, I don't know. It's a very interesting question that I'm interested in. But you can imagine that if you have too much myelin, that can be a problem if now the signals are no longer synchronized.
Okay, thank you.
You're welcome.
Any other question? We may still have some time. Yes, please.
Is it still? Oh, okay, perfect. Throughout the different presentations, there was emphasis on the fact that structure and function are really related in different manners. But from time to time, we could see also that there was variability between where certain functions are situated compared to some certain gyri or sulci. And also, when we do registration of brains to a template, usually we only use, well, most of the time we only use the structure of the brain. And I was wondering, because I've seen a few articles doing functional registration, but do you think this is something that should be more widespread and should become a new standard, or we should still keep doing registration mostly on the structure of the brain?
Yeah, I think I can answer. So I guess just for some context, a good example of uniting structure and function in the registration process is the Human Connectome Project. They collect a bunch of data, including resting state data, and use that in their their generation of what's called the FSLR template, which is similar to the FreeSurfer template, but obviously uses this other functional data as well. My intuition, having used it and FreeSurfer's registration, which is structural only, is that they're pretty similar for most research, but they're not identical, and that the FSLR template is better for certain things. I can't comment as to whether it's always better. I just know it's better for some things. But my intuition about the question of whether we should continue using structure or structure and function is that you use them for different things. If you want to isolate structural variability, you want to align using FreeSurfer. If you want to get maximum alignment between subjects, you would probably want to align using both function and structure.
So, you know, I'm a post-mortem person, so I'm going to ask a follow-up question to this. What about the single subject analysis? I mean, in the past, from what I know, functional imaging studies concentrated on creating the average, and that was what they said. This is the area involved in whatever function. And now I think the trend is moving more towards doing the actual analysis in the individual brain space. So that gives you a better correlation between the activation and the anatomy of that subject. Because the point that you were making concerning there's a different location of the activation focus with relation to a macroanatomical landmark. It was so funny when you were presenting your video, I thought, oh, look at that, the brain seems to be changing more in the occipital pole than in the frontal pole. I wonder if that is something like generalizable, or is it just because you're presenting data from the visual system, so of course the color changes are happening in the visual system?
Yes, that's a great question, and actually, I haven't calculated anything that would say, this part of cortex is the most changing between subjects, but my intuition is actually that the occipital cortex is probably the most changing between subjects, or the most different between subjects. And the reason for that is largely that when we look at the occipital cortex, first of all, in that animation, you were seeing real differences. The occipital cortex does change a lot more between subjects, and what we found is that, I think I said in the talk, we can have variability of almost 4 times in terms of magnitude of just the size of V1, the first visual area, whereas cortex only varies by maybe 1.5 times across subjects. So that variability in the occipital cortex has to be made up by stability somewhere else. And it's possible that auditory cortex and sensory motor cortex, for example, are equally variable, or that there are some parts of frontal cortex that are even more variable, but my intuition, at least, is that occipital cortex is one of the most variable. Yeah.
Fascinating. In macaque monkeys, the highest degree of variability, this structural variability, is also found in the occipital lobe. Funny, I wouldn't have thought that. If you think that when we're moving from non-human primates to primates, actually the greatest degree of change happened in the prefrontal cortex. And then it's sort of like, it was surprising for me to see that actually then within humans, there's a greater degree of individual variability in the occipital.
Well, and something that actually deepens the mystery a little bit is that most people's intuition, if you just ask them, is that the first visual area, like the first stop for information from the eyes on cortex, would be less variable than the next one, and that as you go higher up the visual hierarchy, up to like face areas, for example, you would have more variability. But we actually find the opposite. We find that the most variability is in primary visual cortex. V2 and V3, which are around primary visual cortex, have lower variability, and as you go higher, it seems to get even smaller. So it almost seems like the source of the variability might be the visual world that you experience, but I'm speculating a bit here.
Right, because you haven't done, or do you know of studies that have done this kind of analysis, but for the auditory or sensory system? Like, would it be something specific for humans? Because as Cassandra said, you know, we humans, we use our visual system to well, and our hands, you know, but we're basically, we're visual beings. Whereas, for example, rodents would use their whiskers to explore the environment. So in rodents, you'd expect this kind of variability in the somatosensory system, and because we are visual, you have it in the visual system.
Yeah, absolutely. I mean, visual cortex is already a little over 25% of cortex, so it's the biggest sort of, if we think of occipital cortex as like one unit of cortex, it's probably the biggest single unit. So that it makes sense that the most variability would probably also be found there for humans.
Hi, can you hear me? Yep. Well, thanks everybody for the talks. They were all super interesting and fascinating. This is just a very small follow-up comment on what Noah Benson was just commenting on the variability in V1 and downstream areas. And I was wondering, I'm not aware of those studies, but my question relates to it's clear that I also encountered that the variability in the size of V1 is like really remarkably large. I know that that also happens in macaques. Now my question is, I've also encountered quite a bit of variability further downstream too. So my question is if you have you can clarify under what kind of criteria you're making this decision that further downstream is actually more similar compared to kind of earlier areas.
Yeah, so we did a study of the Human Connectome project, which had about 200 subjects who participated in visual mapping experiments. And we actually hand drew boundaries for V1, V2, V3, and are on the process of drawing them for higher visual areas. And we just calculated the coefficient of variation of the size across areas, and that's very clearly decreasing as you go up the hierarchy, or as you go downstream.
Okay, that's interesting. But there might be other kind of criteria, for example, under which, for example, you can say one subject does not have a specific kind of selective area, and then you might think, okay, then it cannot be more different than that because everybody has V1. So it's interesting that maybe different criteria can kind of describe different that kind of variability.
Sure. For example, you might have downstream areas whose location are more variable, but whose size is very static, for example. Yeah, that's a good question. That's something we haven't looked at particularly. Yeah.
Cool. Thank you.
One thing I can add is I think there is old paper in 1997 by Andrews and they published, I think it's Jamie or I think they're doing some post-mortem section to identify V1 based on genomic slide. Then they also found a very large variability in V1. It's about maybe small person, large person, with 3 times differences. So I think it's not variability more clearly specific to neurogenic data as well. Any other questions, comments? Yes.
Again, a follow-up to what variability in lower versus high regions. Do you think this could be related to the task that's at hand? Like if you're maybe doing a grading discrimination or something, maybe that depends more on your lower level, like visual skill that's learned over the course of life, whereas if you have like a naturalistic movie or something like that, which depends less on the skill, but more on the subjective perception, you might see more variability in higher level regions. Would that make sense?
Yeah, so that's a good point, and this is something we absolutely need to look into. It's possible that the stimulus, which is really, honestly, our stimulus is mostly designed for V1. Part of that is that there's only a vague notion of what higher order visual areas are doing. We have a sense of what V2 and V3 are doing, but we don't have a model of it. That said, all of these visual areas do have what we call retinotopic maps, where the whole visual field gets represented in the area. And so we can use these stimuli to draw the visual area boundaries that pretty much tile cortex. However, I totally agree that there's, we've seen in some areas, such as the IPS regions, that the task, whether you have a task that requires you to attend to the stimulus or attend to the fixation dot, will change the appearance of the maps. So we know some of that happens in higher order areas. We just don't have a very good sense of how much, and I think that in order to answer a lot of those questions, we need better models of what higher visual areas are doing. Yeah.
Thank you.
Mm-hmm.
Hello? So in the beginning of the session, you spoke about how people used to argue over drawing boundaries between brain areas and such, and you just mentioned how you're doing this, you're drawing boundaries between brain areas. And I was just wondering, how do you kind of ensure that you're getting the right boundaries? Do you have to have, 10 people doing it and then take the average, or do you do one and have someone else check it, or how does that work?
Yeah, so we've been drawing boundaries for V1, V2, and V3, and we're working on higher order ones. Interestingly enough, in V1, V2, and V3, human raters agree with each other very well, and the main reason for that is because the boundaries are quite clear, typically, in V1, V2, and V3. That said, even as we were working on those boundaries, we realized that there were features in the V3 map that weren't really explained by the standard model of the retinotopic organization in early visual cortex, and so none of this is all that easy or clear. And in V1, there's been a lot of work done on V1. We have drawn it in thousands of subjects, really, across all the research that's been done, and we have a pretty good idea of where it is and what it does, and or approximately what it does and how big it is and that kind of thing. But you get very far into the visual hierarchy, and those, you can talk to experts and they'll be able to draw the maps pretty reliably, but they won't necessarily agree with other experts all the time. For certain areas, we're very good at it. For other areas, we're just not. There's a lot of idiosyncrasy in the maps in like lateral occipital cortex, for example, and I would basically just say that this is an active area of research, and we're still trying to formalize how these maps are arranged and whether there are real individual differences between subjects in their sort of topology. Yeah. Yes.